Thank you guys for having me and showing up, and I hope you guys find this as interesting as I do. So just like Brooke said, I have a PhD proposal defense on Wednesday. What that means is I haven't actually done my entire dissertation. I haven't done my entire PhD. I'm about halfway through, but I need to show my committee members, who are all listed right there, that what I propose to do in my dissertation is actually feasible and will actually get some significant results, and it contributes something meaningful to the scientific field. So that's what I'll be giving to you guys today. So my dissertation title is tentatively titled Longitudinal Biomarker and Chemical Analyses in Marine Vertebrates. And so my committee is, the chair is Dr. Stephen Trumbull, that's Brooke and my advisor. And then we also have 4 other committee members in different expertise that help contribute different components to the PhD. So they help me along in my PhD and make sure my data and my study is a well-rounded project. So the overall goal, and I'll kind of explain all this different terminology as I go through, but is to utilize chronologically growing tissues of marine vertebrates to look at trends in steroid hormones and a contaminant over long-term time periods, which are over a year, and determine how these hormone profiles and these different trends are impacted by the environment and possibly diet. So the vertebrates, the marine vertebrates I'm going to be talking about today are the Yelloweye rockfish, and then I'm going to give you guys kind of a truncated version of this talk, and I'm going to leave out the leopard seal just because of a time limiting factor. So I'm still actually perfecting this talk as we speak. So starting with chapters, so your PhD is broken up into 3 separate research projects, and they're usually different chapters. And so I have 3 different chapters. The first one is longitudinal trends in hormones and development of reproductive parameters of a long-lived teleost. So that's the Yelloweye rockfish. So we'll start with that one. The objectives of that chapter, so what I'm going to try and accomplish, is measure and validate annual hormone profiles of Yelloweye rockfish by measuring these different reproductive hormones, which are progesterone and estradiol, and then also a biomarker of stress, which is cortisol, in the opercula, which is a bony structure that covers the gills, in those annual growth increments. And then from these hormone profiles, I want to help develop a method to assign age of sexual maturity and determine the frequency of spawning events based on reproductive hormone trends among these female Yelloweye rockfish. Okay, so starting with the first objective, I have a hypothesis. So I believe that I propose that steroid hormones are deposited as a function of time in the Yelloweye rockfish opercula in the visible and yellow substrate regions. So briefly, you'll see here, there's yellow stuff right here. You can see right here, there's light and dark growth banding. I'll talk more about those in a second. But I believe that hormones are deposited in each of these layers as a function of time, so over the lifetime of the animal. And I'll, again, get into that right now. So a little background, why would I want to do, why do we care about looking at reproductive hormones and stress hormones in fish? Well, if you think about it, we rely on fish a lot, right? Everyone eats fish. We have commercial fisheries. You go out and you fish for, out on fishing tours, right? So there's a lot of interest in conserving our marine fish populations. And so how we do this, and by we, I mean government agencies such as NOAA, they calculate what's called stock assessments. And so these stock assessments are based upon what's called the maximum sustainable yield. And so it's just a way of calculating how much fish we can actually remove from the ocean without depleting the stock, okay? And so to actually come up and create these stock assessments every year of each different fish species and the different populations within those species, you need a lot of data, okay? So You need different types of data such as catch data, so how much fish are we actually removing from the oceans? We need data on the abundance. How much of the fish are actually in the ocean? What are the numbers? And then we also need biological data. And this is where my research kind of comes in. And so we need different information on reproduction. And so we need how many females are in the population of the different fish species we're looking at. How often do they produce? How much do they reproduce? All these different things are really important in terms of building this stock assessment and coming up with accurate maximum sustainable yields. And so based on the availability and the amount of data that we have on each different fish species and the different populations are placed into what's called tiers for management purposes. So 1 through 6. So if we have a lot of information and we have really reliable, accurate estimates of this maximum sustainable yield, you're in tier 1. If we know nothing about your fish species population, and really we just come up with this maximum sustainable yield based on the amount of fish we catch every year, you're placed in tier 6. So we don't know anything about those fish, right? And so you use these different data based on catch, abundance, and biology to create these different models which help predict the maximum sustainable yield. And so these data also help determine what's called the total allowable catch. And so if we look at this figure here, this is the amount of fishing, right? So the catch of tons of a stock, right? So if we're fishing and we have really reliable data, we can come up with a total allowable catch. And then also the acceptable biological catch. Those are the two amounts or catch limits we set to say, as long as we stay within these catch limits, we're not depleting the fish population. Now, if we go into what's called the overfishing limit, then we do know that we're actually depleting the stock. But in order to actually measure and calculate an accurate estimate of these, we need all these different types of data. Okay. So now going back to the Yelloweye rockfish. The Yelloweye rockfish is found throughout the Pacific Northwest. It is a human-targeted fish species, so we do catch them commercially. They're not as big of a fishery as, let's say, salmon, but they do have a fishery for them. And they are placed in the tier 4 and tier 6. And remember, tier 4, we kind of know some data about their life history, some things about their biology, the catch abundance. Tier 6, we don't know really anything, okay? Anything reliable. So they're also threatened in Puget Sound due to overfishing. So in Puget Sound, they're listed as threatened under the Endangered Species Act. And part of the reason why they're so susceptible to overfishing is because of their slow growth and maturation. So their max age is around 100 years or over 100 years. So they can live a really long time. Also, they take a long time to sexually mature. And so the actual age of sexual maturity is approximately 22 years old. So if you think about it compared to humans, we go through puberty around our teens, right? Well, these fish take a lot longer. They take about 10 years longer. And so they take a long time to sexually mature. And if you're overfishing at a really fast rate, these populations aren't able to replenish the fish that we're taking out as quickly as we're removing them. And then another interesting thing about the Yelloweye rockfish reproduction is that they are viviparous reproducers. So that means they give birth to live young. They don't lay eggs. They have internal fertilization by males, and they give birth to live young. Whereas most fish are oviparous, so if you think of the salmon that spends its whole life swimming up the river, the female deposits all these eggs at the riverbed, and then the males come in and spray all their sperm. That's external fertilization. These fish don't do that. They have actually a gestation period, and they'll actually give birth. The benefits to this is that when they do give birth, the larvae are larger than, let's say, a salmon egg that's laid. They're larger, more developed, and so they can swim. They have better sensory development, and so they're a little bit better off when they're actually let off into the ocean compared to oviparous reproductive. So in terms of the annual life or reproductive cycle of the yelloweye rockfish, they actually have oocytes. Those are the eggs. They mature within their ovaries over the course of the winter season. And so that's about 5 months. And then they become fertilized in the spring by the males, and then they finally give birth in the summer. And they are thought to reproduce annually, so they are thought to go through this process every single year. And also, like I said, it takes them a long time to mature. They grow to pretty big sizes. And we found that with these rockfish species, larger females produce more young. And so this is what this figure is showing. On the y-axis, you have fecundity, so that's the amount of larvae that a female can produce. And you see that as the female weight increases, they're able to produce more young. So if we take out and overfish and take out these large females, we're disproportionately affecting the amount of reproduction, like resiliency to overfishing if we take out these large females. Okay, so now how do we study and kind of how can we track the reproduction of the yelloweye rockfish, okay? Well, let me walk you through a process that's called vitellogenesis. So we have our rockfish here, and then the ovaries of the female are right here. They're developing, and this yellow egg-shaped thing are the eggs. And so let's say this is time increasing from left to right. Now, vitellogenesis is a process that's vital to the development of fish eggs. And so what it does is vitellogenesis produces what's called vitellogenin, which is a lipoprotein. It's a nutrient. And that vitellogenin is transported from the liver down to the growing oocytes. And so that's what all this yellow stuff is in this schematic, okay? That's all that vitellogenin. And so what controls that process is a hormone called estradiol, okay? So as these oocytes begin to grow, a steroid hormone, which is estradiol, begins to increase, and that's released from the ovaries. And then we also have a steroid hormone called progesterone. And so this hormone is also helping develop the final maturation stage of the egg. So it's finally ready to become fertilized by the sperm. And so both of these steroid hormones are really important players in the development of the oocytes in the ovaries. Okay. And so another hormone that I mentioned is cortisol. And why would we look at cortisol? Well, if a fish encounters a stressor, and so if we define stress as any deviation from homeostasis, the internal body functioning of the fish, that can be termed a stressor. And so for females, this could be reproduction. Developing lots of eggs is a really stressful process for the female fish, right? So what's going to happen is the brain, there's two types of stress response. There's almost the instantaneous stress response, which happens with the neural detection of this stressor, which then stimulates what's called the chromaffin cells, and these are located in the kidney of the fish. And the chromaffin cells are going to release what's called catecholamines. And those are other hormones, like epinephrine. And so epinephrine, what that's going to do is kind of perfuse the gills with blood. So it's sending all the blood to the gills so that it can become oxygenated and provide more energy for the fish to deal with the stressor. A more delayed, sustained stress response, though, is the secondary response that happens about minutes and sometimes even hours after this initial stress response. And that's when a hormone called corticotropin releasing hormone from the brain, from the hypothalamus, stimulates the pituitary gland and releases what's called adrenocorticotropin hormone. And that travels to what's called the intrarenal cells, and that's also located in the kidney. And that's going to release cortisol. And so cortisol's main job is to delegate energetic stores from the body to deal with the stressor, which in this case could be reproduction. Now, it also could be chronic stressors, such as starvation. So if the fish is chronically starving, cortisol is elevated for a very long period of time, and that can have some negative effects as well. So how can we track lifetime reproduction and stress in fish? So that's where these chronologically grown tissues come into play. And so what I mean by chronologically grown tissues is just that these are tissues that over time, they lay down sequential growth increments, okay? And so they're constantly growing throughout the lifetime of the animal. One you may be familiar with is called the otolith. That's the fish ear bone. And so all fish have ear bones. And so this is a yelloweye rockfish otolith right here. And you can see that they get this banding, the light, dark growth layers. And this is similar to tree rings. As a tree grows, it lays down light, dark growth layers. So does this otolith, okay? And so what you can do is actually count the number of pairs of these growth layer groups, and you can get the age of the fish. And so this figure is actually showing you that we get 19 growth layer bands here in the otolith. And then here is the fish opercula, so that structure that covers the bone. And we see that there's the same amount of growth layer groups in the opercula as the otolith. So we know that these opercula lay down these growth layer groups in the same manner as the otolith. Okay? And so what that means is if we can extract these reproductive and stress hormones, we can save a lot of time and cost. So let's say if we wanted to measure the hormones of a wild fish over its lifetime, we'd have to go out every single year and catch that same fish. It's not really that feasible. And then also, we can do what's called a baseline correction. So your normal circulating concentrations of these hormones, you always have some sort of concentration flowing through you right now. You have progesterone, estradiol, cortisol flowing through you right now. But you're not stressed. You're not, like for females, if you're not pregnant, those hormones aren't going to really change, right? So that's what's called a baseline. But these baselines can shift. As you grow older, as you become sexually mature, your baseline will increase. And so a good example of this is if I catch a fish, I measure the stress hormone cortisol in its serum, in its blood, and it's 1,000 picograms. And I catch another fish, and that fish's cortisol concentration in the blood is 500 picograms. You would honestly say, okay, well, this fish that I first caught with the 1,000 picograms is twice as, could be possibly 2 times more stressed than this other fish. When in reality, that's not actually, you don't actually know that because it could be just different baseline concentrations of hormones. That fish could be bigger, that fish could have had different genetics, come from a different gene line. And so all these different factors come into creating your baseline. And so what we can do is we can actually determine what the baseline concentrations are throughout measuring these annual hormone concentrations. And we can take that into consideration when we look at actual changes in these hormones. And so this also provides retrospective studies. So we all have heard about climate change and how the Earth is rapidly changing, especially in the polar regions like Alaska. And we want to know how these animals are going to fare with changes in environment. Well, we can go back in time. Remember, these fish live about 100 years. And so we can go back 100 years and see what their physiology was like then, possibly before all this climate change started to occur, and then see how their physiology has changed. Okay, so if we blow up the opercula, so this is how it would situate on a fish, we see that they do have this banding here, which is the visible region. My graphics aren't showing up, that's all right. But then we also have this yellow region, which is what we call that YS region. And so to actually extract the hormones from these growth layers, we had to go out, and by we, I mean I have collaborators at Alaska Department of Fish and Game. One of my committee members, Dr. Dion Oxman, he's actually one of the co-PIs with my advisor on this project, Dr. Stephen Trumbull at Baylor. So we have a lot of collaboration on this project. And so they're able to go, and they have connections with NOAA, and NOAA goes out on what's called long line surveys. And these long line surveys go out, and all they do is catch fish. And it's a way to sample, get that data for that abundance that you need for those stock assessments, right? And so they do this every single year at all these different locations, okay? So this is where we get our samples from, kind of central, more eastern Gulf of Alaska. And we can get, from this, we also get their opercula, and then we also get their age, their length, weight, and maturity status. So we know if they're growing eggs within their ovaries, or if they're not, or if they're immature. Okay? So luckily, Alaska Fish and Game, our collaborators, they actually clean the opercula for us, and they count all the growth layers in the opercula for us, so I don't have to do that, so I'm very appreciative of that. So when we get the opercula down at Baylor, we can actually sample these growth layers in the opercula. And so we developed a sampling scheme to actually sample the yellow substrate region. So if you look at this figure, you can see that this is the visible growth layers. Obviously, it's not as pretty as those other opercula I showed you. But then we get this huge, large YS region. And so how do we sample that? Well, based on younger animals and the visible growth layers we can see in the younger animals with the opercula, we do know that about from age 1 to 19, the growth layers are about a 1 millimeter wide. And then after that, they slow down their growth, and they're about a 0.5 millimeter wide. So this is kind of the sampling paradigm we came up with. So up until this would be the age that corresponds to 19, and we sample at 0.5 millimeter growth, the widths, and then we kind of expand that to 1 millimeter. And so we approximate that that's about a year's worth of growth. So this is kind of what it looks like. You sit and you can sample these growth layers with nail clippers and then surgical scissors. You literally will sit there with reader glasses that are like 2 times magnifying power, and you sit there and you clip out or you cut out these growth layers layer by layer until you can see here the entire part of the opercula is sampled. And so to actually extract the hormones from these growth layers, we use what's called a SOX-TEC extraction of the steroid hormones. So this is a pretty common method. We use what's called a 2-to-1 chloroform organic solvent in combination with heat. And so we're pretty much boiling out all these lipids and fats from the bone tissue, and they dissolve into the organic solvent, okay? And so steroid hormones are lipophilic, so that means they're found usually with fats and other lipids. So we know if we're extracting all these lipids and all the fats, we're most likely getting the hormones as well within these growth layers. And then we actually measure them using what's called enzyme-linked immunoabsorbent assays. And so each hormone has a kit from a company that is specifically designed to measure those hormones, those specific hormones. And this schematic here on the right is just showing you the concept. It's called what's called a competition competitive binding enzyme assay. All that means is when we add our sample, so the extract from each growth layer into a well, which is a 96 plate well, and we add in a competitor to bind to the bottom of these wells that are predetermined to only bind, or they should only bind to progesterone or estradiol or whatever hormone we want to look at. If we have a lot of hormone in our sample, we add a reagent to change the color throughout this enzyme or this ELISA process. And the amount of color change actually correlates to the amount of hormone in our sample. So if we have a lot of binding to these, the bottom of the plates, so that only bind to progesterone, then the color change that we see here will be, it'll be really light. So there's no color change. And so that means we have a lot of hormone in our sample. But if the color change is a lot, so it turns really dark yellow, so that's the color it'll turn for our enzymes, then that means we don't have a lot of hormone in our sample because that competitor bound to all the available spots on the bottom of the cell. It's a pretty common method of determining these hormones, and so it's a pretty, also that means it's a reliable way of measuring these hormones. And so this is just the statistical analysis that I'm doing. And so for analytical validations, all linearity tells you is that the kit that we use to measure these hormones and the tissue we extract them from, there's no interference. So we are measuring the right amount of hormone within our tissue. And then accuracy is making sure that our kit that we use can reliably measure really low concentrations of that hormone in our sample to really high concentrations. So it's making sure that that kit, the number we get from that process or that ELISA kit is actually accurate. And so we're also doing physiological validations. And so we're looking at the variability among hormone profiles, using what's called an annual lifetime Z-score profile. All that is we're kind of normalizing all the data we collect from one animal to its average lifetime concentration of each hormone. And I'll kind of show you that in a second. And then we use what's called repeated measures, ANOVAs, to just compare the hormones we get in the visible and the YS regions to make sure that they're deposited in a similar way and that the hormones aren't deposited differently in the YS region compared to the visible region. And so we have preliminary results. This is just straightforward that the linearity and accuracy test all passed. So we did all the necessary tests for that. That just means our kits are correctly measuring the right hormone and in the right concentration. Now to the cool stuff. You can see here, this is on the y-axis, your hormone concentrations on the y-axis, and then the age of the fish on the x-axis. And this is from an 18-year-old female and a plot of a 42-year-old female. And so each of these data points is an average year-long concentration of that specific hormone. And so one thing I want you guys to take from this is that you can see from 0 to 5, this fish had relatively higher hormone concentrations compared to this 42-year-old. That, I think, gives a good example of that change in baseline. So just because we measure higher hormones in this fish doesn't mean that it is more stressed than this fish. It could just mean that this fish had different life history events happen to it compared to this other fish. So again, showing that kind of benefit of looking at these long term. And then you can obviously see that around age 15, we get these large spikes and oscillations. And so, especially with this 42-year-old, something crazy happens at the age of 20. Okay? And then we see that as it gets older, the annual concentrations increase. But we have to kind of see the magnitude of those changes. So are those changes relatively large or are they small? And that's where that Z-score comes in. And so what we do is we normalize it to the annual lifetime concentration of these hormones for each individual fish. And so what that means is if you have a Z-score of 0, the 0 is the lifetime average, right? Okay. If you have a number above 0, that means for that year, that fish sustained, on average, higher hormone concentrations than it has throughout its entire life, okay? And so we can see that these peaks are falling above and below this 0 line, okay? And especially for this 42-year-old, we see at age 20, something crazy happens where all these hormones spike. And then at age 35, they continuously increase. And so this is achieving objective A and showing that we validated these kits and the measurement of the hormones within these growth layers. The Z-score analysis shows that we are getting oscillations, so the variability along the opercula indicates that these are biologically relevant hormone concentrations deposited through time. If all we saw was one concentration throughout the entire lifetime of the animal, this wouldn't really be useful, right? But we are seeing these increases and decreases. And so we can potentially construct a method for developing reproductive parameters, which is objective B. And so my hypothesis is that the first peak we see from baseline in progesterone and estradiol will occur during the estimated age of sexual maturity in yelloweye rockfish. So that'll be on average from 11 to 22 years old, okay? So that's the range at which these fish become sexually mature. And then also it is predicted that peaks in progesterone and estradiol from baseline following the age of sexual maturity will correspond to a potential spawning event. And from that, we can use those to calculate what's called spawning frequency. So the methods for this are pretty straightforward. The first observed Z-scores, so those Z-score profiles I showed you, were both estradiol and progesterone. Remember, those are the reproductive hormones. As long as those are both greater than the average lifetime hormone concentration of that fish, we can reliably infer that that's possibly the age of sexual maturity. This figure here kind of shows you that what Ateki et al. Did in 2003 is they took all these immature rainbow trout and they injected them with E2, which is estradiol, and then also progesterone. So they had never reproduced at all. They had never developed oocytes in their ovaries. And they found that this actually stimulated what's called an increase in the luteinizing hormone. Okay? And so luteinizing hormone is a key player in stimulating the activity of the ovaries. So we do know that progesterone and estradiol can both influence the activity of the ovaries. And so that's just saying that not only do they play a part in developing eggs, but also activating the actual ovaries for the first time. And then spawning frequency is just the sum of the peaks of progesterone and estradiol, where the Z-score is greater than 0, divided by the total reproductive years. So that's all the years after sexual maturity are potentially reproductive years where these fish can spawn. Again, we have to kind of ground truth these a little bit. I've kind of already explained why Z-score greater than 0 would be an appropriate measure for these different parameters. And then traditional outlier analysis may be inappropriate due to biological application. And so what that means is usually you want traditional identifying of outliers is, okay, so a data point 2 standard deviations above the mean would be classified as an outlier. However, what that means in terms of our study is that a fish would have to sustain over an entire year on average 2 standard deviations above its annual lifetime average, which physiologically probably wouldn't happen because these hormones are only activated for certain periods of the month, right? So 5 months, 6 months, and then the other 6 months they're potentially dropped, right? So we actually have some ground truthing in this, in that the ovaries that have been examined from the opercula we have in the lab showed that we have 4 fish where they had mature ovaries, so that means they had developed oocytes within their ovaries. And if we look at the most recent growth layer and the progesterone and estradiol concentrations, they're all greater than the average lifetime hormone concentration. So that they're all over that 0, right? And then we have this one fish that had both progesterone and estradiol skyrocket, and they had what's called resting ovaries, and so that means there was nothing in there. And I think that this fish potentially had already spawned by the time that it was caught. So it had higher hormones in that first year. So getting to the actual results of implementing this method, this is a 38-year-old fish. And we see that up until around age 20, we get these significant deviations, or deviations above the average lifetime concentration. And so the estimated age of sexual maturity is within that range from the published literature. And then it also has an estimated spawning frequency of 52%. So in any given year, this female would spawn on average 52% of the time, so almost 50-50. So this significance is showing that we have determined a potential way to actually calculate the age of sexual maturity and spawning frequency in female yelloweye rockfish. So both of these parameters are both key variables for those models that I was talking about in the beginning with the stock assessments. But can we go further? So can we see how these parameters and these hormone concentrations change with environmental variables? Which leads me to my second chapter, which is the long-term environmental impacts on the reproductive and stress physiology of a teleost. And so this objective is just to determine how these hormone profiles and these reproductive parameters, age of sexual maturity, and spawning frequency relate to long-term changes in environmental patterns. So both of my hypotheses are that they will be associated or correlated, both the hormone profiles and these parameters will be correlated with long-term environmental changes. So long-term environmental changes, what does that mean? There are certain different processes in the atmospheric processes that influence sea surface temperature and productivity within the oceans. And so if we look at these different processes that do affect those things, one of them is what's called Pacific Decadal Oscillation. And so this is on average a 20 to 30 year change in sea surface temperatures in the Pacific Northwest, or the Pacific North Pacific, greater than 20 degrees north. And so you can see here, that's what this figure is showing. So on average, when you have a positive index of these values, you have warmer sea surface temperatures. And then if you have cooler or negative values for this index, you get cooler water temperatures. And so this is all related to El Nino events. And then we also have the Northern Pacific Decadal Oscillation. So this is another index that's really similar to the PDO. This index has been correlated with changes in productivity, so changes in phytoplankton and zooplankton biomasses. So when you have an increase in that index, you have it's been related to more productivity in the ocean. And this is based on sea surface height variability. And if we look at the northern, what's called the Northern Oscillation Index, this deals with sea level pressure anomalies. So as winds or as air from the tropics become warmer, they rise and they travel to the north and they become cooler and they drop down, and that affects different sea level pressures. And that correlates with the Backrun Upwelling Index. And so upwelling is associated again with primary and secondary productivity. So as wind is blowing across the ocean along the coastal zones, it actually forces water to move away from the coast. And we have deeper water that comes up from the surface to replace it. And that usually has more nutrients available for the phytoplankton and zooplankton to actually increase in biomass. And so what we actually see is that Yelloweye rockfish growth has been correlated with all of these different indices. But it's interesting because this study by Black et al. Has found that populations in Bowie, which is at like the base of the southeast Alaska, and then populations in California have inverse relationships with sea surface temperature. So fish in Alaska grow larger or have faster growth with warmer water temperatures, and in California it's actually cooler temperatures, they have greater growth. So it's kind of interesting. We don't really want to predict what is going to affect what. So the methods for determining how are we going to see how these long-term environmental variables impact these hormone profiles and reproductive parameters. Well, we're going to use all the same data, all the same stuff I talked about in the first chapter, and we're going to create a model using all these different environmental variables, and all these variables are available online, so you guys can all go out and check out these data if you'd like. Our hormone data follow what's called a Poisson distribution. Again, this is more statistics, and all that means is that we have a positive skew to our data, so they're not normally distributed. So we just have to adjust our modeling for that. And we use what's called a general linear mixed modeling approach. And so all I'll say about this is that we have to correct for this Poisson distribution, so that's why we use a general linear approach. And then we also have to take into account for the Yelloweye rockfish ID. So each Yelloweye rockfish, and I showed you that, for each individual we have numerous observations, okay? And so all those observations are potentially related to each other. And so that would introduce a bias to the models. And so to correct for that, we use what's called a random effect. And so we use what's called the lowest AICC score to determine the best model. And then we do the same thing with the reproductive parameters. We use all the environmental variables as what's called fixed factors. We take into account the Yelloweye, the repeated sampling of the individual Yelloweye rockfish. And again, we use what's called the lowest AICC score for the best method, or the best model. And so I will just show you a couple. I haven't done this yet. Like I said, it's a proposal. So all I'll show you is the plots of the z-scores for all the fish I have. So these are mean z-scores for the different, plotted on the y-axis for the 3 different hormones against the different environmental variables on the x-axis. So we have sea surface temperature here, and this dashed line is at 0. So remember, anything above that, that's higher. Anything below it is lower, or anything below it is on average lower hormone concentrations. This is the Pacific Decadal Oscillation, the Northern Oscillation Index, and you can see if it centers around and it doesn't really do anything, there may not be a huge relationship with that variable. We have the North Pacific Gyre Oscillation, the Bakun Upwelling Index, and then finally sea surface temperature anomalies. And so the one thing that I want you guys to take away from this is that when we do see warmer temperatures, we do see an increase, it seems, of all the hormones. And then we also see with an increase in sea surface temperature anomalies, so that's deviations from normal sea surface temperatures. When that deviation gets above 0.8 degrees, we also see these hormones start to increase. And remember, if we go back to the growth with the Southeast Alaskan rockfish, they were all positively, they had growth positively correlated with sea surface temperature, warm sea surface temperatures. So the significance of this is that we can determine which environmental variables positively or negatively impact lifetime reproductive and stress hormones among the female Yelloweye rockfish. And then we can also understand how past environments have impacted the age of sexual maturity and spawning frequency. And so understanding that will be able to help us forecast into the future how these fish will fare with warmer temperatures that are bound to happen in Alaska. And so with that, I will leave you with that, and thank you. And if you guys have any questions, you can ask them now.